This video demonstrates the dissection of mouse retina and subsequent acquisition of single-cell suction recordings for cones. The eyeballs of a euthanized, dark-adapted mouse are cauterized at the dorsal part and denucleated under dim red light. Eyeballs are then hemisected along the equator. The cornea and ventral parts of the eyeballs are then removed. The dorsal retina is isolated from the pigment epithelium layer and sliced into small, thin pieces using a razor blade. The slices of retina are then placed in the recording chamber that fits to a microscope stage. Several photoreceptor cell bodies are drawn into the recording electrode. When a cone photoreceptor is drawn in the electrode, a bright test flash will induce photoresponse with amplitude above 4 picoamps.
Hi, I am Jin-Shan Wang from Dr. Vladimir Kefalov's laboratory in the Department of Ophthalmology and Visual Sciences at Washington University in St. Louis. Today, we will show you a procedure for recording photoresponses from single mouse cone photoreceptors. We use this procedure in our laboratory to study the adaptation properties of mammalian cones, the photoreceptors that mediate our daytime vision. So, let's get started.
One day prior to the recording experiment, dark-adapt a mouse by placing it in a light-tight cage overnight. Use a micropipette puller to fabricate recording electrodes from borosilicate glass tubing. Under a compound microscope, use a heating filament